For my local expert project, I have chosen to focus on anti-CRISPR. In nature, anti-CRISPR is a system that bacteriophages have evolved and developed to combat the CRISPR systems that they run into in bacteria that so often hinder their survival. The anti-CRISPR system blocks the CRISPR-Cas system from binding to its target gene within the bacteriophage genome using the ACR protein. In biotechnology, scientists take advantage of anti-CRISPR's natural role in the outside world to block the CRISPR-Cas genome editing system in human cells and to restrict editing activity to specific tissues or developmental stages to improve upon the safety and efficiency of the CRISPR-Cas genome editing system. The ACR genes, also known as the anti-CRISPR genes, are the genes that encode for the inhibitor proteins that block the CRISPR-Cas system from binding to its target gene. The ACR gene is often located directly upstream of the anti-CRISPR associated gene, also known as the ACA gene. This association between the two different genes allows for us to locate and discover even more ACR genes in an effort to further understand the many mechanisms of inhibition that the anti-CRISPR system utilizes. As previously mentioned, there are many mechanisms of inhibition that the anti-CRISPR system employs, and this figure displays 3 of them. In the figure on the slide, the researchers show the production of the cascade complex complete with the assembly of the mature CRISPR RNA before inhibition. The fully assembled cascade complex is then inhibited in one of 3 ways in this example. The inhibitor protein ACRF2 binds to one of the cascade proteins that helps make up the cascade complex. Another inhibitor protein called ACRF1 binds to 2 completely different cascade proteins on the complex instead of 1. And the final inhibitor protein shown, called ACRF3, binds directly to the Cas nuclease that cleaves the target DNA and as a result, physically blocks the nuclease from actually cleaving the DNA. These are only 3 examples of how the anti-CRISPR system inhibits the cascade complex, but researchers are convinced that there are many more strategies that have yet to be discovered. So how does learning about anti-CRISPR tie into these last 2 modules? As everyone should already know, the CRISPR-Cas system is used to study genomic variants and gene knockouts. When this method is combined with transcriptomic analyses, we can also measure the effects of gene alteration. The anti-CRISPR system can be utilized to significantly improve the CRISPR-Cas specificity. It can block the CRISPR-Cas system from targeting a specific gene, or it can restrict editing activity to specific tissues or developmental stages. This allows for fewer off-target effects that could impact results from transcriptomic and genomic analyses to occur. Since anti-CRISPR and Module 2 both deal with transcriptomics and genomics, this procedure is tied into the second module. Despite their usefulness, there are some limitations to anti-CRISPR. To start off, as mentioned beforehand, there are many strategies of cascade complex inhibition that we have not discovered yet, and the reason for this is because there are so many ACR proteins due to each one's specificity for a specific sequence. Thus, it is possible for anti-CRISPR to not be an option in the laboratory depending on what the sequence is and if we have already discovered the corresponding ACR protein already. In addition to this, we will need to perform more experiments to determine if ACR proteins are safe and effective. We currently don't know if they induce toxicity, provoke a host response, or halt gene drives. Some other techniques that achieve the same goal as anti-CRISPR include creating a better guide RNA with higher specificity for its target gene, engineering the CRISPR-Cas system itself to be more specific and efficient, or using viral vectors in combination with the CRISPR-Cas system to make CRISPR-Cas more specific. Despite the existence of other techniques that could improve the specificity of the CRISPR-Cas system, the anti-CRISPR still holds many advantages over all of these. With engineering the guide RNA and Cas protein to be more specific, we would have to tailor each guide RNA and Cas protein to each individual target gene. This would cause a lot more work for the researcher than using an anti-CRISPR system, because even though I mentioned beforehand that each ACR protein is specific for an individual gene, researchers have just begun to slowly discover that there are a few ACR genes that work for multiple target genes, and there might be even more that haven't been discovered. With the viral vectors lack cell specificity, so there could be off-target effects with the viral vectors. However, because of how specific anti-CRISPR is, there is a very little chance of off-target effects using anti-CRISPR. The anti-controls that would most likely be in use would be the following. If possible, you will want a control that includes the anti-CRISPR system with the CRISPR-Cas system, using the same ACR gene as in your experiment with a different target gene than the target gene being used in your experiment, with results that you know that you are expecting. This will allow us to know whether or not the anti-CRISPR system is working. The result we would expect to see would be one that is known from previous research or experience. The second control is a sample without any genes but with only the anti-CRISPR system, with only the reagents and CRISPR-Cas system. This will allow us to determine if there are any contaminants with our reagents. We would expect to see nothing. The third control is that of just the CRISPR-Cas system and its target gene. This will allow us to determine if the CRISPR-Cas system is working. We would expect to see the splice gene. The final control would just be the target gene to make sure that it is in there and that there are no mutations or contamination created in the preparation of the gene. We would expect to just see the overall target gene. This figure comes from the paper, An Anti-CRISPR Protein Disables Type 5 Cas12a by Acetylation. The goal of this research was to study the newly discovered anti-CRISPR protein, ACR-VA5, and report the mechanisms by which it inhibits CRISPR-Cas12a. During this research process, the researchers found that ACR-VA5 acts as an acetyltransferase. To verify this, the researchers determined the crystal structure of ACR-VA5 and superimposed it upon the crystal structure of another acetyltransferase called NATD. This is all shown in Figure A. The structural alignment and structural homology between the two structures further corroborate the claim that ACR-VA5 is an acetyltransferase. To further prove this claim, the researchers studied the interaction between ACR-VA5 and acetyl coenzyme A, as shown in Figure B. The bent conformation when bound to the acetyl coenzyme A further proves that ACR-VA5 is an acetyltransferase. Based on all of this information, the researchers predicted that if there were mutations at the K2A, K29A, G32R, G34R, S35A, Y57A, and S74A positions, then this would impair ACR-VA5's ability to recognize acetyl coenzyme A, which the researchers have predicted is important for ACR-VA5 to act as an anti-CRISPR system and inhibit CRISPR-Cas12a earlier in the paper. Figure C shows the results of a cleavage inhibition assay. In other words, the figure shows whether or not ACR-VA5 is able to act as an anti-CRISPR system to inhibit CRISPR-Cas12a from cleaving the target DNA sequence from the remainder of the gene. Mutations of each of the positions recognized earlier that can impair ACR-VA5's ability to recognize acetyl coenzyme A and thus impair ACR-VA5's ability to inhibit CRISPR-Cas12a was put in each lane. ACR-VA5, CRISPR-Cas12a, CRISPR-RNA, and the double-stranded DNA with the target gene for CRISPR-Cas12a were put all together in a buffer system to observe whether or not there would be cleavage reactions with each mutation. This allows for the researchers to determine which mutation is important for ACR-VA5's ability to recognize acetyl coenzyme A and act as an anti-CRISPR system. The gel allowed for the separation of the substrate two cleavage products if CRISPR-Cas12a was not inhibited, CRISPR-RNA, and processed CRISPR-RNA. There were three different controls. The no anti-control included everything except for the anti-CRISPR system and ACRVA5 to ensure that CRISPR-Cas12a was working. The wild type included everything including wild type or non-mutated ACRVA5 to ensure that ACRVA5 was working. The final control was the input control, which included just the target DNA and CRISPR RNA to ensure that the target DNA and CRISPR RNA were in there and that there was no contamination by reagents. All three controls showed the expected results, confirming no contamination or errors in preparing the assay. In this assay, the researchers saw cleavage products for all mutations, no matter how weak the bands. This confirmed that all of the positions in ACRVA5 play some kind of role in recognizing acetylcoenzyme A and inhibiting CRISPR-Cas12a. The stronger bands for each cleavage product showed which ACRVA5 position is more important for anti-CRISPR activity. The paper, Anti-CRISPR ACR-IF9 functions by inducing the CRISPR-Cas complex to bind DNA nonspecifically, focuses on an ACR called ACR-IF9. More specifically, the researchers investigated whether or not ACR-IF9 inhibits the CRISPR-Cas complex by misdirecting the complex to bind to nonspecific DNA. The nonspecific DNA doesn't contain a PAM sequence or a complementary DNA sequence. In order to carry this out, the ACR-IF9 must actually physically bind to a type IF CRISPR-Cas complex to misdirect it to a nonspecific DNA sequence. This complex with ACR-IF9 bound to it will from here on out be called the CSY-F9 complex. A fluorescence polarization or FP assay was used to study the DNA binding properties of CSY-F9. There were three types of double-stranded DNA, and all double-stranded DNA were fluorescently labeled for detection and measurement. The three types of double-stranded DNA were nonspecific double-stranded DNA, which didn't contain any complementary binding sites for the CSY complex, specific double-stranded DNA, which contained a complementary binding site for the CSY complex, as a regular target gene for the CSY complex would, and double-stranded DNA 2X, which contained two 24 base pair complementary binding sites for the CSY complex. In Figure A, the researchers measured the binding of fluorescently labeled specific double-stranded DNA and non-specific double-stranded DNA to CSY and CSY-F9 complexes as the complex concentration increased. The amount of fluorescence reflected the amount of binding that occurred. In Figure A, the controls were the CSY and specific double-stranded DNA and the CSY and non-specific double-stranded DNA. As expected, the researchers saw that the CSY complex was bound to the specific double-stranded DNA sample, and that the CSY complex was not bound to the non-specific double-stranded DNA sample. This confirmed that the CSY complex alone was working, and that the specific and non-specific double-stranded DNA samples were not contaminated. The researchers then observed if and how strongly the CSY-F9 complex would bind to both types of double-stranded DNA. The researchers saw that the CSY-F9 complex bound more strongly to the non-specific double-stranded DNA than the specific double-stranded DNA, confirming that the role of ACR-IF9 is to misdirect the IF CRISPR-Cas complex to a non-specific sequence to act as an anti-CRISPR system. Jumping to Figure C, we see the same exact FP assay being run, except with the specific double-stranded DNA and 2X double-stranded DNA. The researchers investigated the difference between the interactions between CSY and 2X double-stranded DNA and the specific double-stranded DNA, and that between CSY-F9 and 2X double-stranded DNA and specific double-stranded DNA. The researchers expected and verified that if the double-stranded DNA had two specific binding sites for the CSY complex, then there would be 2 times the binding between CSY and 2X double-stranded DNA than CSY and specific double-stranded DNA. However, we saw that with the CSY-F9 complex, there was no change in the amount of binding between the double-stranded DNA sequences and the CSY-F9 complex, no matter how many binding sites were available. This again confirms that the role of ACR-IF9 is to misdirect the IF CRISPR-Cas complex to a nonspecific sequence as the number of specific binding sites for the CSY complex doesn't change the amount of binding that occurs. Figures B and D are both EMSA gels that display the different protein complexes detected. In Figure B, the researchers detected the amount of 2x CSY bound to the 2x double-stranded DNA, the amount of 1x CSY bound to the 2x double-stranded DNA, and the amount of free DNA. In this gel, we see that as the concentration of CSY grows, the amount of bound CSY to 2x double-stranded DNA grows. 2x CSY bound to 2x double-stranded DNA has a greater presence when compared to 1x CSY bound to 2x double-stranded DNA at 6,000 nanometers of CSY. And 1x CSY bound to 2x double-stranded DNA has a greater presence when compared to 2x CSY bound to 2x double-stranded DNA at 125 nanometers of CSY to 4,000 nanometers of CSY. This signifies that when we do have enough of CSY at 6,000 nanometers to have a ratio of 2x CSY to 2x double-stranded DNA, then the 2x CSY bound to 2x double-stranded DNA is far more prevalent than 1x CSY bound to 2x double-stranded DNA. Thus, this figure proves the affinity between CSY to the target DNA. We know that the EMSA gel worked because when we look at the 2x double-stranded DNA lane only, we only see free DNA, meaning that there is no contamination and that the EMSA was run successfully. In Figure D, we now focus on the CSY F9 complex. We see that there are basically only CSY and CSY F9 bound to DNA rather than CSY bound to DNA, showing that the CSY F9 complex is most likely successfully binding to the DNA and misdirecting it, and outcompeting with the regular CSY complex for the target DNA. This supports ACR-IF9's success as an anti-CRISPR system. Again, in the double-stranded DNA lane only, we only see free DNA, and in the CSY-F9 complex only, we only see the CSY-F9 complex, meaning that there is no contamination and that the EMSA was run successfully. The study Strong and Turnable Anti-CRISPR Cas activities in plants focused on the characterizations of two anti-CRISPR proteins, ACR2A4 and ACRVA1 in the plant, Nicotiana benthamiana. The researchers also demonstrated that ACR2A4 and ACRVA1 prevent site-directed mutagenesis in leaves when transiently co-expressed with CRISPR-Cas9 and CRISPR-Cas12a mediated gene editing respectively. In the figure on the slide, the ACR2A4 was specifically focused on. The researchers wanted to analyze the ability of ACR2A4 to prevent dCas-EV2.1 mediated gene activation of two endogenous N-benthamiana genes, NBDFR and NBAN2. dCas-EV2.1 is a Cas9-based PTA or programmable transcriptional activator that activates CRISPR. Thus, in this case, the researchers are analyzing the ability of ACR2A4 to act as an anti-CRISPR, because if ACR2A4 can prevent dCas-EV2.1 mediated gene activation of NBDFR and NBAN2, then we know that ACR2A4 can prevent CRISPR-Cas9 from acting. In Figure A, the researchers measured the relative NBDFR gene expression for 4 different samples. The NBAN2 gene, or GNBAN2 with ACR2A4, or NLS-ACR2A4, GNBAN2 alone, the NBDFR gene with ACR2A4, and the NBDFR gene alone. QRT-PCR of each sample was taken to evaluate the relative NBDFR gene expression of all 4 different samples. In Figure A, we see that the only time we see the expression of the NBDFR gene is when there is only NBDFR gene in the sample. This is expected because the NBAN2 gene sample acts as a control. We expect to see no expression of NBDFR because there is no NBDFR in the sample. Thus, if we don't see any relative NBDFR gene expression, then we know that there is no contamination and that the QRT-PCR was successful. We also expect to see no expression of NBDFR in the GNBAN2 and ACR2A4 sample because there was no NBDFR genes in the sample. And we expect to see no expression of NBDFR in the GNBDFR and ACR24A sample because the ACR24A sample is expected to inhibit the CRISPR-Cas9 system so that NBDFR is not expressed. Thus, we see that ACR24A does indeed successfully act as an anti-CRISPR system. The GNBDFR acts as another control, and we expect to see expression, which means that there is no contamination of the sample and that the qRT-PCR was successful. Figure B corroborates the same conclusion that ACR24A acts as a successful anti-CRISPR system against CRISPR-Cas9 because of the significantly higher expression of GNBAN2, which is now the gene being studied rather than the NBDFR gene, when compared to the remainder of the samples. The main difference between Figure B and Figure A is that there was expression of the NBAN2 gene in all samples, including the samples in which we would expect no gene expression, which are the GNBDFR and ACR24A and GNBDFR samples. There should be no gene expression under these two samples because there should be no NBAN2 genes even present. Thus, this suggests that there was contamination in this experiment. This would also explain the reason why there was relative NBAN2 gene expression in the GNBAN2 and ACR2A4 sample. The relative NBAN2 gene expression was about at the same level as the controls that should have shown no relative NBAN2 gene expression. And so based on the normalization between all of these samples, we know that ACR2A4 and GNBAN2 would have most likely shown no NBAN2 gene expression had there been no contamination. Thus, we know that ACR2A4 was successful as an anti-CRISPR system.